Hi, everyone. We're going to get started for today. I wanted to welcome you all to our exciting webinar about our Vevo F2, Empowering Researchers of Ultrasound Technology. My name is Christina Asa. I'm a Scientific Applications Specialist with FujiFilm VisualSonics. And today our presenter is our very own Andrew Needles. A couple of notes about today's webinar. We will make a recording of the webinar available afterwards for anyone who was not able to attend today. All lines will be muted for the duration of the webinar, but I do encourage you to please use the Q&A button at the bottom of your Zoom panel at any point throughout the presentation to ask any questions you might have. Questions will be answered by Andrew at the end of the session today. We expect our session to be approximately 40 minutes, leaving us 10 to 15 minutes for questions. Without further ado, our presenter today is Andrew Needles. He's been with FujiFilm VisualSonics for more than 10 years. Over that time, he has held many roles from engineering and product management to director of marketing and product management. In that time, he's been integral in the developing and launching of several of our products, including, and most recently, the Vevo F2, which he will be speaking about today. So without further ado, I'm going to pass things over to Andrew.
Well, thank you very much, Christina. It's a pleasure to be online with all of you today. I'm presenting here from my home office. I hope all of you are staying well, healthy, safe, and again, we appreciate you taking some time today to join us here online. So let me get into this presentation. This is a really new and exciting technology. We've partnered for this webinar with the IEEE Society to hopefully introduce this to a number of new people, and we're very much looking forward to sharing this with you today. A little bit about our company, our purpose and vision. It's really about bold innovation and empowering those that are dedicated to the advancement of human health. This is our mission and purpose and vision. Our platform, the Vevo F2, is really the next step in driving the future of translational research to meet our customer needs. For those that may not be aware of some of our current products, I just wanted to introduce you to some other products here in our portfolio. We do primarily focus on the preclinical imaging market. So this is really focused on imaging of small animals in human disease, for human disease models. Here we've got an ultrasound system called the Vevo 3100. This is an ultra high frequency imaging system that's capable of imaging at frequencies up to 70 megahertz. We can also couple a laser, near-infrared laser component with the system to combine to what's called the Vevo Laser X. This is a photoacoustic capable system for doing molecular imaging in small animal species. The systems are, as I mentioned, ultra high frequency, which means ultra high resolution. We can also then use those in some particular clinical applications, and that brings us to the Vevo MD. This is a system that is used in clinical research. It has full FDA clearance and Health Canada as well, CE marked in Europe and other regions of the world. This system really addresses the clinical needs of our researchers. However, there was an area which all of you are very familiar with, that we did intend to address, and that is really where the Vevo F2 comes in. It is primarily geared towards acoustics and engineering type research. As mentioned, these are researchers, many of them like yourselves, who are studying ultrasound technology or becoming interested in ultrasound technology. And this system really enables all of the very high resolution imaging that we've always offered, but also access to very, very low level tools that enable the collection of very low level channel data. And I'm going to show you more about that and how it all works today. So this is just to set the stage of the various different markets and segments that we do cater to. Again, the preclinical space, the clinical space, and now moving more into the acoustics and engineering space as well. So we are looking to expand our reach. What you can expect to find from the Vevo F2 are features that will satisfy the imaging needs of acoustics researchers, ultrasound engineers, and also users that are needing both the combination of ultra high to low frequency imaging capabilities. And this is the Vevo F2. With the Vevo F2, you can expect a highly flexible imaging system. This is flexible primarily in its ability to go from 71 megahertz all the way down to 1 megahertz, covering a broad range of imaging species, imaging depths. The architecture is very open. We provide access to the pre-beamformed individual channel data. This is something in a mode of imaging and data collection called VADA, the Vevo Advanced Data Acquisition, and I'll share much more detail about this mode to you shortly. It's very intuitive. As you can see, there's a touchscreen display. It's easy to use with a graphical interface, highly configurable, and very, very usable. We now have one system that's adaptable for imaging at a variety of imaging depths, both at ultra high frequencies where it's much more superficial, going all the way down to lower frequencies to get much deeper through the tissue for deeper imaging depths. And it is photoacoustic capable as well. So I mentioned the Vevo Laser X. The Vevo Laser X component, the laser component, is now compatible with this for multimodal imaging. So these are some of the key features of this new platform. There are several other things to point out. The system does come equipped with standard Bmode imaging. I mentioned the Vevo Advanced Data Acquisition. New to our platforms is the addition of a triple transducer connector. This allows us to connect multiple transducers at the same time to the system. We have a touchscreen user interface, as mentioned. The electronics now support all of our standard 256 element linear arrays. We do support phased arrays and dual frequency arrays, some of which are currently in development, and I'll share a few examples of those with you shortly. And finally, our parent company, Sonosite, does produce low frequency clinical grade transducers, also used in veterinary applications as well. We partnered with Sonosite to adapt some of their low frequency array transducers onto our Vevo F2. These would typically be compatible with their export system. So this is really a big advancement for us, the ability to add these low frequency arrays in partnership with our parent company, Sonosite. Just to dive a little deeper into some of the details here, you can see, as mentioned, the graphical user interface, all of the touch screen control panel, a beautiful display monitor here that gives striking images. We also have built-in on board something known as our Vevo HD image technology. This is a image processing technique which produces highly enhanced image visualization to go along with our inherent high resolution imaging. A lot of the data can be taken off the system as well and managed in our Vevo Lab environment. Here's the triple transducer connector I mentioned. 3 transducers can be connected. One transducer will be active at any given time and can be selected via the touchscreen interface. The pulse configurations, and we'll get more into this, but everything from waveform voltage and delay profile can be controlled at a very, very low level for any given transducer. As mentioned, it is photoacoustic capable, and a number of the key specifications, clearly the frequency range, 71 down to 1 megahertz. The system is capable of ultra-fast imaging, over 50,000 frames per second. It does use a true Nyquist sampling rate up to 192 megahertz, ADC resolution up to 12 bits, and time delay resolutions on the transmit side of 2.6 nanoseconds, and on the receive side, time delay resolutions of 0.65 nanoseconds. So these are very, very precise delays which are driven by these high frequency electronics and allow us to do this very high precision beam forming and image formation, which I'll show you some examples of shortly. Here's a fairly long list of all the different transducers and possible uses. In vivo, you can see some of the in vivo applications ranging from things like mouse embryology all the way down through cardiovascular imaging. We're getting into rats, larger rats. These are the high frequency transducers which are used for a variety of different applications in the frequency ranges up above 10 megahertz all the way up to 70. You can see that as we go down in frequency, we do get deeper into the body and vice versa. Or into the tissue, if we go up in frequency, we see much higher resolution, but with more limited depth. Here are 2 of the low frequency transducers that are a product of our parent company, FujiFilm VisualSonics. The L38XP, this is a linear array in the 10 to 5 megahertz range. Again, more suitable for larger animals, larger rats and rabbits, potentially. This also has the capability of doing low frequency photoacoustic imaging. And then the phased array, which as we go into the future, we hope will be more applicable for some cardiovascular applications as well. Sorry. This is just a visual to help give you a sense of the imaging depths and resolution that we're talking about. Here we're looking at a series of phantom images. This is using just a standard cyst phantom, and you can see here with the L38XP, the scale bar here is 5 millimeters, so we're fairly deep through the tissue mimicking material. If we take a region of interest and just zoom in, now taking the UHF-22X, this is the 22 to 10 megahertz, we can see much clearer resolution and delineation of these regions of the cyst. However, we are starting to lose some of the imaging depth. If we zoom in even further, now we're looking at the UHF-57X. This is all the way up to 57 megahertz, starting to see much more detail around these inclusions within the phantom. However, we are getting some more attenuation at these higher frequencies, and the imaging depth is now starting to be limited. So again, this trade-off between depth and resolution. And at the very, very highest, we can start to see very detailed structures within that inclusion itself, imaging just almost about a centimeter here in depth at the very highest frequencies. So again, just to give you a sense of the scale of what we're talking about as we range from the lower frequencies, sort of 5 megahertz range, all the way up to 71 megahertz. I'd like to get into more detail now about the Vevo Advanced Data Acquisition. This is really going to be important for anyone who's interested in getting individual channel data off of our arrays that are connected to the transducer arrays that are connected to the system. This allows the ability to do offline beamforming, image processing techniques, new and advanced methods, and I'll show you some of those here in the next little bit. There is a highly usable graphical interface that goes along with this mode, making it very straightforward to get up and running with your data collection. This slide just illustrates the various portions of the transmit signal chain on an ultrasound system. So here's our transducer. It's showing the animal here, a mouse or a rat perhaps in this case, being imaged. And we can see that from the user interface, we have the ability now to set up things like transmit and receive configuration. We have our transmit pulsar and beamformer. All of these sections along the chain can be configured through the VADA interface. So we can define our pulse sequences. This allows us to set up the sequence of transmit events. We can configure the shape and timing of the ultrasound pulse in a given channel. We can also set the amplitude using the voltage rail control. And then finally, we can set our transmit delays to allow us to set specific focusing patterns, or also allows the ability to select a plane wave if that is something you desire. So lots of control over the transmit side of the signal path. Looking at the receive path, we can see that as the signals come in on the front end of the electronics through the analog digital converter, the individual raw channel data can now be acquired. This is the new element on the Vevo F2 that was not available on previous generations of our system. We still then do the beamforming, envelope detection, scan, conversion to ultimately produce an image. There are various data points still available as have been on our previous systems, some of the other platforms that I showed you earlier. So we can get beamformed RF data, we can get a raw data, which is an envelope detected version. We can get all of the various images and cine loops off of the system. All of that's still available. What's new here is really the ability to get down to the individual channel data, which is very, very important if you are experimenting with beamform techniques, plane wave imaging approaches. So how does this all come together? This is the user interface that I had mentioned earlier. Here we're looking at the transmit delay panel, what we're looking at is a steered 5-degree steered plane wave. So we can see there's no focus pattern. The time delays here are really just set up to give that 5-degree angling of the transmit profile. We could also set up something similar. You can see examples, focused beams as well, and that allows us to really set up particular transmit delays that we desire. We can then start to program in waveforms. Here we just see a simple single cycle pulse, but there's lots of flexibility in terms of how long the pulse you would like it to be, the polarity, whether you'd like to invert the pulse, the amplitude. We could set up sequences of these waveforms as well. So that leads us to the next tab, where this is where you really start putting it all together. Everything is now done via this flow chart method here, where we have start and end. You can see there's a Bmode ping, the aperture loop is set up. All of this then puts together a sequence of pulses and waveforms to allow you to collect all of that data. And finally, you can start to preview what that might look like. Here's an example looking on element 36 of the array. This is just from a wire target. And then you can also then start to look across all of your elements. And we see here, this is an example of element 91, again, collected from a wire target. You can see the echo off the wire target. So again, almost very much like an oscilloscope at this stage, which allows you to get very, very deep into the data and see what's going on at a channel level. I would encourage you, if there's more interest in really taking a deeper dive through the VADA interface, I would encourage you to please reach out, during this call or afterwards. Please do connect with us, and we'd be happy to set up some time to really walk you through on an individual basis in more detail to understand some of your needs and exactly how the VADA interface could be applied for your data collection. So what are some of the things people may want to do using this interface? Well, plane wave implementation I've mentioned several times. This is probably one of the biggest requests that we do hear from users and potential users of our instruments. Here we're looking at typically ultra-fast imaging techniques, where we are firing all elements simultaneously, creating that plane wave. That's what allows us to get up to those ultra-fast frame rates that I mentioned earlier on, upwards of 50,000 frames per second. So some of these techniques allow applications such as Ultrafast Doppler, super resolution ultrasound, and other techniques that are new and emerging modes of acquisition in the ultrasound field today. Next is beamforming itself. So plane wave, in some sense, is a method of beamforming, but just to develop new algorithms, you do need access to those individual channels to bring them all together and produce those high quality ultrasound beams, which ultimately lead to these high quality images. So this allows you to test novel beamforming techniques for image reconstruction. This is another big area of research that VADA could be used for. If you are working with external devices, for example, high intensity focused ultrasound, where you're using higher amplitude pulses for doing therapeutic applications, perhaps shear wave imaging, generation of elastography, or acoustic radiation force imaging, anywhere where you're using an external device and you need to synchronize that with your imaging sequences, the VADA interface is equipped and set up to allow you to do those kinds of external syncing and coordinating between multiple pieces of equipment. And last but not least is the ability to image small to large animals. So especially for studies that are looking at not just imaging, but also tissue characterization, really trying to get to the underlying RF data, the VADA interface does allow you to do that type of data collection using both small to large animal models on one single platform. So these are some of the target areas. As we go forward, we are exploring other things as well. I've mentioned plane wave imaging, ultrafast Doppler, signal processing, beamforming, tissue characterization, super resolution imaging. And something I'll show you a little bit about today is a technique called dual frequency imaging. So these are all areas of research that are ongoing in the ultrasound field today, and things that we are now exploring with the Vevo F2. we're doing a lot of that exploration with a variety of key opinion leaders in the field. These are some of our early adopters and key collaborators working with the new technology. Professor Stuart Foster from Sunnybrook Health Sciences Center at the University of Toronto. His main application area really is dual frequency imaging, and I'll show you a few examples of that shortly. Professor Jean-Luc Genesson from France, again, super resolution imaging, elastography, ultrafast Doppler are his areas of research. One of our earliest users, Professor Magnus Synthio from Lund University in Sweden, he's investigating plane wave imaging. Again, ultrafast Doppler, you can see that's a common theme. And his research is really aimed at translational research. They are actually users of the Vevo MD technology as well in clinical applications, also looking at photoacoustics in clinical translational applications as well. And finally, Professor Gerhard Schmitz from the University of Bochum in Germany. He's highly focused on super resolution imaging and contrast imaging. We've been collaborating with his group on our Vevo 3100 system, and now beginning work on the Vevo F2 as well. So these are some of the key leaders that are really helping us drive new applications, new methods on this new platform. A little bit of work from Stuart's group in Toronto. This is, again, so apologize for the math here, but just to give you a sense of what we call conventional beamforming. I've mentioned beamforming a few times, but I'm sure most of you are familiar, but if not, really what we're talking about is a series of ultrasound elements. This is the so-called array that we talk about. And as we're firing ultrasound waves, essentially we're solving a geometric problem, trying to understand how long did a particular wavefront from a particular element travel, and then making those corrections such that when we get back to the source on the get back to the receive side and bring those signals into the system, we can use these geometric equations to correct for those time delays and essentially form our image. This is a nice example from Stuart's group, really just validating the VADA interface and all of the beamforming that they're doing here using that approach. This is the standard Bmode image that comes out of the system. Here we've got a series of wire targets providing a nice pattern. We can use this to really verify that our own beamforming techniques on the system itself are working. So this is just a standard Bmode image of a phantom that you would see on the system display. What they have now done, and this is work done by Jackie Caminiti at University of Toronto, working in Stuart's group. She's taken this data using the VADA interface, applied all of the beamforming approaches, and essentially validated that what's happening on the system itself is accurate, and then applying some additional windowing and apodization, getting even a nicer, cleaner image of these wire targets. One thing I just wanted to point out here about Jackie's actually has come to Stuart's lab as a summer student, and I think it really is a testament to the work that our engineering team put into this user interface, because literally within a couple of weeks and a few conversations with us, Jackie was able to get all of this up and running using the VADA interface and put together a really nice proof of concept here of validating some of the beamforming. So this was really nice to see that someone so new to the field could essentially get up and running, doing some fairly complex mathematics and produce some really nice images here. So I should point out, apologize, just come back. This is actually done with that L38 XP transducer, so you get a sense of the depth in the phantom. We're well over, we're almost 5 to 6 centimeters in this case, or 60 millimeters deep. So much deeper than we're used to on our with our higher frequency transducers. So that this is really nice to see as well. So conventional beamforming, that's fine. That's fairly well known, and those last images really validated what we are seeing on the system and the VADA interface itself. But what about plane waves? Well, plane waves again are really, in essence, a method of transforming these delay profiles that are flat. They can be steered, and then trying to back propagate and reconstruct what would have been the original source. So this was some hydrophone measurements done with low frequency plane waves. And you can see here, using a hydrophone measurement in a water tank, we can start to measure various angles where we, again, would have used the VADA interface to set up those transmit sequences I showed you earlier. So again, just to give you a sense of the different angles, again, this is done by Jing Yang, who's a student at Stewart's lab at Sunnybrook. So again, similar mathematics, a little bit different because now we have these flat waves. We can steer them as well. This is just to give you a visual of, again, you're trying to solve these geometric problems once the waves come back. And what we can see here, again, some work done by Jackie, that this, again, our standard Bmode image, when we collect the VADA data, this is what it looks like right out of the, just the raw data essentially that comes back. And you can see it's individual channels. There's been no beamforming applied. So you see this somewhat, messy image that's hard to interpret what you're looking at. Well, when you then start to apply all of those mathematics, and also what's happened here is applied 31 different angles and compounded them, you start to produce a plane wave image that's very, very similar to what you would have expected to see using standard Bmode. So this is a nice demonstration of how plane wave imaging, especially with this angular compounding, can actually produce an image that's quite equivalent to standard Bmode. What you're not seeing here, because this is a still image, is that those plane waves can be fired much, much more rapidly. So the net effect is a much, much higher frame rate image. And this is when we get into the ultrafast imaging, which is why there's such interest in these approaches of imaging now, because you can essentially get the high quality image that you like and improve your overall frame rate in the process. So again, a nice demonstration here, just validating that the VADA interface in use. Just going to shift gears a little bit more into some applications. This is an example of using, I'll show you a couple examples using microbubbles. Again, this work is done in collaboration with Stewart and his lab, and also the group at University of North Carolina, Paul Dayton and the Dayton lab. When we look at microbubbles under ultrasound fields, we can see that the tissue has this very narrow response. If we transmit around 3 megahertz, we get something somewhat in this range. Microbubbles, on the other hand, when we look at them, have this very, very broad frequency range. And what we'd like to do is essentially detect bubbles in the region where we don't have tissue, so we can specifically isolate them. Here's an example of a microbubble oscillating in an acoustic field. And here's actually some real data that was measured from the microbubble's response. So what I'm going to show you next is an approach called dual frequency imaging. And essentially what dual frequency is trying to do is, with a single transducer, excite at a low frequency, image at a high frequency. And these are the kinds of things that you can start to do. This was a publication from several years ago. You can see, obviously, Stewart and Paul, part of this publication. This was actually looking at some molecular imaging, but they essentially, what they've pioneered here is an approach called acoustic angiography. And the reason you're seeing these are tumors in an animal model, and you're seeing some molecular imaging here, and targeted imaging. But if you focus on the vasculature, these are really exquisite images. And this is all a function of the fact that, if I just go back here, we've been able to image much higher in the frequency range. We've excited at a low frequency where the bubbles want to be struck, and that's where they have the most resonance, and then detecting at a much higher. I apologize, I don't have a picture of the transducer. I should have added that, but there is a customized transducer that was developed as part of this research project, essentially to combine low and high frequencies. So this is active research that's ongoing. And where this leads to the Vevo F2 is we are now starting to test these transducer devices using the F2 platform. We're actually using at this moment two systems, because we've got one controlling transmit, one controlling receive. You can see we're transmitting down at 3 megahertz, imaging up at up to 40 megahertz using a linear array. This is just using a flow phantom. Very early images, some of the first images that Jill's just collected, showing that contrast agent flowing through the tube detected here. Getting some fairly good contrast tissue ratio of over 20 dB, which is actually very, very good. So again, the goal here is to continue to integrate this solution into, so we would essentially be running off one platform. But these are some of the kinds of things that are starting to be developed. So this is all work that's going on with the group at Sunnybrook. Just one final example. This is work that's been done in collaboration with the Schmitz Lab in Bochum, Germany. This is, again, an example using a tumor model. This is an approach called super resolution imaging, also a contrast-based approach, but not dual frequency. This is just done at a single frequency, and it's essentially a method of bubble tracking. Now, these images were, in fact, collected with our Vevo 3100 system, but I did want to show you some of these new results that we're getting just using native frame rates on the order of about 50 frames per second on our Vevo 3100. The work that's ongoing now, and hopefully we'll be able to update you in the near future, is to integrate some of this on the Vevo F2, where we expect even better sensitivity and detection as we improve those frame rates and make use of those ultra-fast frame rates that I was commenting on. So even here, we're getting highly detailed images of the microvasculature within these tumors, far better than anything we'd see simply with Doppler imaging or one of our more standard approaches. But again, this is another nice approach to doing contrast imaging in these tumors. So again, these are just a few of the applications. I just really wanted to say thank you to all of our collaborators and our KOLs that we've been working with. We'll continue to develop more and update on more applications as they're developed in the future. So just to summarize our VADA interface, VADA, I hope I've demonstrated for you, it is an advanced set of graphical tools. Really enables the custom configuration of waveforms, pulse sequences, delays, profiles, all of that. So whether you're doing dual frequency imaging, super resolution imaging, conventional beamforming approaches, non-conventional approaches, all of this is possible now as the interface and allows you to collect that individual channel data and really analyze both pre and post beamforming. There are areas that we continue to develop. This product is launched. We launched the product in May of this year, but we continue to develop, and you'll be seeing more improvements coming as we go forward. One of them is integrating more system programming and interface and application programming interface on the system itself. That's something that we've heard feedback on and would like to do more there. Also adding more image processing into the system itself, so an environment that would allow for more advanced image processing. And the dual frequency, we feel, is a really interesting application. I showed you one example with microbubbles, but there are other potential uses, everything from lower frequency photoacoustic imaging, elastography imaging, anything where you need the combination of both low and high built into a single transducer device could potentially benefit from dual frequency. So integrating that further so that we are running those imaging methods right on the Vevo F2 and on a single F2 is something that we're continuing to work on and again, collaborating with the group at Sunnybrook on that. I didn't want to forget to mention, I did say it earlier on, but just to remind you that the Vevo F2 is photoacoustic capable. So it supports the addition of this laser X, called the laser X solution, which is essentially the addition of this near infrared laser card. All of the modes of imaging, so the standard PA mode, oxyhemo, which is a method for doing oxygenation imaging and calculations right on the system. The multiplexing features, which allows you to essentially look at different spectral content. And if you're looking at, for example, say contrast agents or multiple contrast agents or endogenous signals in the tissue, such as blood or melanin or fat, by using this multiplexing tool, you can essentially do a spectroscopic version of photoacoustics, which allows you to then segment out different tissues and different reagents that you may have injected into the tissue. This ties in with the spectrum mode and all of the multi-wavelengths. So this laser will actually run from around 700 nanometers well up to just over 2000 nanometers. So you've got a broad range of optical wavelengths that you can use. For those that aren't familiar with photoacoustics, perhaps please reach out and we could discuss this further, but just to make you aware of the fact that the photoacoustics is now available on the Vevo F2 and now available with low frequency imaging as well, which for many photoacoustic applications is quite important. So using that L38XP transducer, we can get down into lower frequency range for photoacoustics. The last thing I would like to share, I showed you all sorts of things about beamforming and dual frequency and new modes of imaging and data collection. Well, I just do want to point out that the system itself is a highly usable and high quality imaging ultrasound device as well. So all of the imaging that VisualSonics is known for at high frequencies and high resolution is still possible. We have we're using the standard Bmode that is built into the system. So when doing any data collection, it's important to be able to find your regions of interest. And that's really what the Bmode, built-in Bmode is for. So here's an example of a mouse kidney. This was done at our up to 57 megahertz. You can see nice structure here within the kidney. It's outlined here. This is about 2 millimeters, so you get a sense of the scale. This is a very, very tiny organ inside a mouse. This is an image at using our highest frequency transducer. This is a mouse carotid. So again, very, very small vessel, certainly less than a millimeter. I don't have the exact measurement, but you can see the scale bar is 2 millimeters. So very, very tiny vessel, and you're seeing that branch of the carotid and very superficial within the mouse. And again, this is done using our highest frequency transducer, the UHF 71X. Image of a mouse heart. This is one of our many very common applications. You can see the UHF 46 and the mouse heart, just over a few millimeters in diameter. In a rat kidney, so now we're going deeper into the tissue, or down at the UHF 22X. You can see the kidney here outlined, the capsule, and then actually the adrenal gland here just off on the right side. And one last example, this is in a diseased liver model. You can see the fatty liver, the normal liver. You can see really the striking difference in terms of the echogenicity of these tissues. Again, this is done with the UHF 22. So just to point out that there is a very high quality Bmode image that you can get on this platform. And then from there, you would dive in and collect all of the data that's required using the VADA interface. That's really the typical workflow. So I did want to share some images with you as well before we wrap up. So see, I'm pretty much just right on schedule here, just a couple of minutes left before I'll be happy to take some questions. But just to summarize, I hope I've been able to demonstrate you that this is really a completely new hardware platform we're dealing with. This is something that we've been working on over the last several years with our R&D teams. The architecture, we really put a lot of effort into making sure that the full access to the channel data was available to support all of the ultrasound and acoustic research needs that are out there. I've showed you the VADA interface with the graphical user interface. It's very user-friendly. It's used to control and configure the system. I didn't mention, but I just have left myself a note here to remind myself to say that in fact, the user interface is underpinned by an XML scripting language. So I know many people in the engineering community prefer to write their own code and maybe work more at that level. That's actually something that is possible. So everything you see on the screen with the user interface is actually underpinned by an underlying XML language as well. So that's something you can that may be important for some users. The hardware is probably the most, I would say, from a technical perspective, the most compelling part of the system. The fact that it supports transducers all the way from 1 up to 71 megahertz. As far as we know, this is the first ever ultrasound to run both low and ultra high frequencies, really getting up to that 71 megahertz range. this is really the first system, ultrasound system in the world that's been able to do this. So we're quite proud of that achievement, and we'd be quite happy to show you more about it and how this can really benefit your research needs. And again, the low frequency transducers, as we partnered with FujiFilm VisualSonics, our parent company, and we'll continue to do that in the future and build out even more low frequency applications with this system. And last but not least, I mentioned the LaserX solution. So for those that are interested in photoacoustic imaging as well, it is available and enables the full range of all of the photoacoustic capabilities that we have available today. So with that, I will stop, and I thank you for all of your attention, and I'd be happy to entertain a few questions. I know we've got a few minutes left here to take some questions. So Christina, I'll pass it back over to you, and let's see what I see. There's been a few questions coming through, so I'll take a peek here and see what sort of things are being asked, and if I'll leave it to you to moderate those questions.
Great, thank you very much, Andrew. That was a really compelling and exciting presentation about this new system that we have to offer. So I want to remind all of our attendees today that we do please encourage you to use the Q&A button at the bottom of your Zoom panel to enter any questions you might have. As Andrew mentioned, we have had a few questions. I'll also mention, sorry, that if we do run out of time, we will be sure to respond to your questions personally by email after the webinar. So Andrew, the first question we have here is, what is the number of independent receive channels in ultrafast imaging? Is it possible to see an example of ultrafast imaging or ultrafast Doppler?
Yeah, great, thank you. So just on the channels, the receive channels on the system are, there's 64 receive channels and 128 transmit channels. The question about the ultrafast Doppler, I don't have any here today, but it's something, it's actually something with our, I mentioned those KOL sites, like we literally have just launched the product. We're working with our KOL, so I would expect in short time we will have some examples. So that's something we'd be happy to follow up with you on to show some examples of how these ultrafast image sequences can be generated. So thank you for that question.
Thank you, Andrew. The next question, there's two I'll combine here about using the Vevo F2 for clinical trials. Is there anything approved as far as human use, or are there plans to do so?
Yeah, great question. It's actually get that question quite often, especially I think with the combination of high and low frequencies. It's a natural thing to want to then think about, okay, how can we use this in humans? So to be clear, it is not cleared for clinical use today. I would say part of our roadmap has always been to try to leverage the technology that we developed for preclinical use or research use into clinical. I think the Vevo MD is a great example of that. We had many major requests over the years to use our technology in the clinic, and as a result of that feedback, we eventually created the Vevo MD. I see something similar happening for our Vevo F2. At this stage, we are primarily focused on more the engineering field, but as these requests continue to come, we will be looking at ways to essentially get a clinical project going to create a clinically cleared and approved version of this. For the time being, if it's something of interest, we can't really as a company market this as a clinical system because it just simply isn't. But what we could do is we could certainly speak with interested people about ways of perhaps using the system under IRB or local ethics approval, doing all of that sort of properly and in a proper way. That's something that could be done and discussed at this stage if there's interest in that.
Thanks, Andrew. We also have a couple questions more centered around the photoacoustic component. So the first is it possible to do 3D imaging in photoacoustic mode?
So the feature set on the Vevo F2 today actually doesn't have 3D built in. It's something we are considering for a future release. So there are, with our other, the Vevo 3100 and LaserX system, there is 3D possible there. So at this stage, there is no 3D, but it's a good question, and I think it's something that we can, if it's important, I mentioned that continued development on this platform, it's something we can certainly look at.
Thanks. And also on the photoacoustic side, the receive chain can the receive chain directly be manipulated for photoacoustics? And is there support for the transmit system in the case of the use of photoacoustics?
It's a good question. I think, I'm just trying to think through this one. I think probably a bit of a detail, but when you get to photoacoustics, because we're using this near-infrared laser, the laser we have has a rep rate of 20 hertz, and that's fairly typical of lasers in that class. So as a result of that, you're naturally working at 20 hertz. And in fact, the photoacoustic data that we collect in the photoacoustics mode is actually at the channel level. And part of that is because the frame rates they're not ultra fast frame rates, they're actually quite low frame rates. So as a result, you actually get you would get all of that channel data in photoacoustics inherently. So you may not actually ultimately need the VADA interface for that. On the transmit side, if you were interested in doing transmit sequences that were different, then you would use VADA. You could use that external syncing feature that I showed you, and essentially set up an imaging sequence which was a combination of Bmode delay profiles, then the photoacoustic receive profiles, and you could essentially collect your data that way. I hope that answers the question, but so the short answer is yes. I probably went a little bit into the method of how, but yes, you could work with the receive data and on the I assume by transmit you're talking about Bmode, because in fact in the photoacoustics there is no ultrasound transmit, it's just a laser light excitation.
Thank you. So switching gears a little bit, what is the ACD sampling rate of RF data, especially for the high frequency transducers?
So the sampling rate goes up to 192 megahertz. That's used when we're at our highest frequencies. Depending on the frequency of the transducer. So for example, as we drop down, I don't have the exact numbers in front of me, but when you drop down to sort of that 30 megahertz range, your sample rates would drop down accordingly. So it's sort of 96 megahertz. So it really depends. The sampling rates are running to be optimal for the transducers that are used, but in principle, the system can sample up to 192 megahertz.
And on the sampling, is it IQ sampling, Nyquist sampling?
It's Nyquist. So yeah, people may be familiar with our older systems where it was IQ. This is actually true Nyquist sampling on the Vevo F2.
Great. And what kind of pulsar does this system use?
That one's it's a good question. If it's okay, I prefer maybe not to. It's getting a little bit into perhaps some proprietary details, but if that's really an important question, I'd be happy to perhaps talk about that offline in more detail. And we might have to talk about some confidentiality agreements or things like that. So I apologize for that one. It's not something we can get into too much detail on this call.
All right. What about someone's inquiring about sharing any results on work that's been done for tissue characterization?
I would say the data we've got today, as you say, we've done a lot of phantom validation. There are publications that we could perhaps try to find from earlier systems. So at this stage, we are really, as we move into the market with this product, this is exactly the kind of thing we're looking to collaborate with users on. So with this system being so new, we don't have a specific tissue characterization example that I could share with you, but that's something that would be very interesting to start looking at. And if there was, I think perhaps for that question, if there's any particular type of data you're looking for, that's actually something where perhaps we could collect some of it and share it with the interested people that are looking to see that stuff.
Okay. Well, Andrew, I think you've done a great job fielding those questions. Again, if we missed any of the questions here, we will follow up with you after the webinar. And if we switch slides here, we want to thank everyone, all the attendees, as well as Andrew for your presentation, for attending today. If I do encourage you to visit our website when you're able to. You can fill out any support forms if you'd like to contact us. Andrew, if you would mind just advancing the slide there. And there's our website right there. We also encourage you to check out our webinar page on our website that has a good library of previous webinars you can check out. Lots of good information there, as well as the ability to sign up for any upcoming webinars. We can be found on all the social media channels, and we will be attending several virtual conferences and have virtual booths at some time. So please go on the webpage there and check out which conferences we will be attending. We also have a learning hub available. So this is for any current users. We have a library of training videos that we've completed and are available for your use and for your assistance. Again, I want to thank everyone so much for taking the time out and attending and having a look at our new Vevo F2 system. And thank you, Andrew, once again for your presentation. Have a nice day.
It was my pleasure. Thank you all. Talk to you soon.